Hello and welcome to the second edition of the VIRTIGATION Plant Health Stories podcast. I'm your host, David Donnerer, Communication Manager for the VIRTIGATION project at the RTDS Group in Vienna. In today's edition, we're going to be looking at the role of the Natural Resources Institute, University of Greenwich. The Natural Resources Institute, University of Greenwich is a UK-based university with a broad history in plant sciences and plant health research, especially focused on the role of whiteflies and the role of climate change in the spread of emerging viral diseases. This is Dr. Svenning Möller, a researcher at NI in the UK, and he will tell us today about his work in the VIRTIGATION project. NI in VIRTIGATION leads the research workstream on the spread of emerging viral diseases under climate change conditions. Could you tell us a bit about the objectives of this research and what approaches you are taking in this regard?
So that's probably, so my main research here at the NOI has been about the host jumping between the tomato leaf curl Nudelli virus going in and becoming a gradually increasing problems. That's basically been the major part of it, along with experiments regarding the resistance to white flies as well, where I assisted with obtaining the cDNA libraries. As my primary work here at the NOI is as a molecular biologist, carrying out those experiments.
Okay. And what are some of the more specific research areas that NI is working on in VIRTIGATION?
So specifically, I've been primarily engaged with figuring out the mechanisms of host jump for the virus part of the VIRTIGATION. So understanding what part of the virus is mutating, how it's going with the plants mutating, and how it's jumping essentially from tomato to cucumber, or rather from cucumber to tomato, or maybe it's doing both ways back and forth. So that has been sort of my primary objective here, has been dealing with that, as well as a whole host of other minor tasks. So that's been my primary objective, and that's been done by swapping out genomes of viruses initially, figuring out first, obtaining the viruses, of course, from leaf samples by sequencing and rolling circle amplification in order to figure out, well, what are actually the viruses, then assessing until we actually had like a virus that's doing tomatoes only, a virus that's doing cucumbers only, a virus that's doing both, and then swapping domains between the viruses and trying to understand, well, which part of the virus is actually responsible for the host jump. And you can think of it like people were thinking around when people had COVID. It's like, well, this thing jumped from whatever, I don't know, maybe some bats into humans. Right. And then some people go like, well, it's because it mutated the binding to that famous lung receptor that meant it went into people's lungs, and then everything happens downstream from that. And so, of course, fortunately for me, we're doing it with like plants, so much safer, and the virus spreads with whiteflies, and these whiteflies are not present in the UK, so it basically can't spread at all. When I'm making, well, doing the lab part of making horrific new viruses by taking out different parts and seeing until I can make a new virus that has a new host range. And that did succeed out. We did manage to identify a particular subregion of the virus that is responsible for this host jump, and as well as coming up with some generalities as to the mechanism for how it's happening. And that's, of course, where basically, in short, is that there are different groups of viruses, some that are infecting cucumbers and tomatoes, and some that are only infecting tomatoes. And in the group that does cucumber and tomatoes, they may have individual mutations that make them lose cucumber infectivity from time to time, or while they lose tomato infectivity. Yeah. So we have different groups which can essentially primarily lose the tomato infectivity, apparently, because it seems that the jump to cucumber requires the acquisition of the DNAB. So the big viruses we're working with are most of them are bipartite, but actually most Gemini viruses are monopartite. So that means you have viruses that consists of one or two genomes. So of course, that's another little thing here is you have a virus, well, and then it has multiple genomes. Well, like a human has 18 different chromosomes, etc. Well, maybe less than that. We have a number of chromosomes, right? And then we have a pair of each of them, etc., etc. For the virus, of course, you might imagine it's just one strand of DNA that's duplicating itself over and over and over to essentially infinite copies within a cell. But when you have a bipartite virus, it has two different components that live together and always must be together that then duplicate themselves up to a number. So the cucumber infecting viruses seems to always be followed with a DNAB component. So they always have the DNAB, and that is presumably then the part that is responsible for infecting the cucumbers. That's
okay. So I think you explained already quite well the mechanisms involved in the host jump. And you also mentioned whiteflies, which I mean are not an issue in the UK, but in other parts of Europe, quite a big issue. And yeah, one of the areas you've been working at NRI is the maintenance of whitefly colonies, which is some complex undertaking. So what techniques have you used to optimize the keeping of whitefly colonies?
well, I mean, so of course, the thing to understand is that whiteflies. So it sounds a bit strange because whiteflies are a problem. Well, can they then be so fragile, etc.? Well, no, not really, because they live in nature, growing on plants, and of course can become a problem. What of course keeps them out of the UK is that there's one thing that instantly kills off whiteflies is very low temperatures. So freezing degrees in the UK will essentially reset and wipe out any population completely. But when you go down to France and you go down particularly to Spain, Italy, they're endemic. And they of course can very easily live over themselves, sucking off plant, placing eggs in the plant, and then rebreeding from those plants. So as long as you keep the temperature control and the humidity reasonably well controlled and keep giving them fresh plants, they're going to keep on living. Now, the actual problem is keeping the cultures consistent because then you have you have a whitefly and then you have there are many different species of whiteflies that have different ability to transfer virus between different plants. Essentially, like they have different preference for plants, they have different preference for virus, and virus have different preference for whiteflies. So they are basically different species of whiteflies. Like you have species of dogs, but the problem is they look exactly the same. So a Middle Eastern or an Asian variant of whiteflies, the big problem you have when working with them is keeping those cultures intact. So because if you contaminate the cultures, you're going to have to go in and, well, start from a single whitefly, sequence in order to see which type of whitefly it actually is you're having. So that is the number one part of maintenance is actually maybe not what people think, but it is keeping very carefully track of who is accessing which colony of whiteflies and keeping them very carefully separated and keeping track of that you have the whiteflies you think you have, because that's one of actually the challenges. The second thing, of course, is just pure maintenance of ensuring that they get fresh aubergines and those aubergines are healthy and that they aren't getting contaminated with virus either, because, well, we don't really have the same viruses, but some of the bigger viruses, once the whitefly has been infected with a virus, it will sort of keep the virus for the rest of its life. They become vociferous whiteflies. So you want to have clean whiteflies and you want to keep track of it, and then you want to basically ensure that they're kept at constant humidity and temperature, and that you check on them and check that they're actually living, they're laying eggs on the plant. Then it takes about 3 weeks or so until you get your they start emerging in mass. They eat the plant. You need to keep transferring them in order to keep them alive, and then do so without contamination from other colonies because they're very small. You might not see them, so you have to be careful with the rooms to keep those cultures pure. And that's pretty much all there is to it. Then, of course, when you do the actual assays, that's the next step of it. It is, well, just having whiteflies is one thing, but having the exact amount of whiteflies on the plant is the next kind of thing of like carefully sucking them up in a tube, essentially, where you filter them out. So we're doing mouth pipetting with whiteflies. It's where it's okay. I mean, insects can be a food source, so it's okay to mouth pipette whiteflies, and you just suck them up. Sort of, they will have some cotton, so you don't accidentally get them all into you. That would be a problem, but you of course want to have the whiteflies stuck inside the tube. So you just move them, and then you move a set number into the plant after you've let them sit a couple of enough days to take up the virus from whatever plant you want to make them vociferous, and then transfer it onto the new plants in order to verify your results that the whitefly is actually transmitting the disease.
Okay, so I mean that sounds quite a complex undertaking. And this, I mean, this research that NI is doing on whiteflies, is it also going to possibly lead to some new approaches to control the whitefly pest, which is more important in other parts of Europe than in UK at the moment?
So what I've been involved in is sort of looking at plants' resistance to whiteflies, because plants have different resistance to whiteflies, and it's sort of a little bit basically putting that's basically both about the experiment of counting, but that was done by another postdoc who was doing the counting with some software of how many whiteflies and then has been transferred to each plant, how many of them survive, how effectively are they living in these different species of cucurbits, cucumber style plants, in order to figure out, okay, which plants are they more or less likely to be on? And then once they're feeding on them, how is the plant responding? Because plants, of course, respond to insect feeding by being a little bit of poisonous to them. Different sugars, different waxes on the plants in order to resist and throw back the insects that's trying to feed on them. And then my role, of course, being a matter of like trying to extract, well, the RNA sequence from those plants, because that's basically a means by which we can figure out which genes is it that's likely involved in this for breeding and creating crops that are more resistant to whiteflies through ordinary breeding means so far, because that's acceptable.
Okay, and one other area I wanted to touch upon is agro-infection, which at NI you've also been working with quite a lot. So maybe you could also explain us a bit your work in that field,
Yeah,
and how it
because
relates to vertigation.
yes, that's the main thing I've been working out with here, is that, of course, to get the virus into the whiteflies, it needs to be in the plant to begin with. And you need to make the infectious clones of the recombinant virus to begin with. And that creates a system where you need to get because the virus, of course, is a difference between the DNA, like getting the DNA into the plant in order to have the virus. It's actually a really fascinating field because, well, it's kind of complicated to explain, because we have the DNA, which is in the virus. So these are DNA viruses, essentially. So just putting the DNA into the plant isn't necessarily going to start up the infection all the time. A particular problem is that when you're doing it with whiteflies, and that's also why we're doing it with whiteflies, to test the results we get and to verify them, that they actually hold up, is that the whiteflies has, like, they come with their own factors when they're stuck on the plant. The whitefly uses the virus and the virus uses the whitefly equally. So the whitefly also benefits from the virus weakening the plant's defenses to the whitefly. Right? And for this reason, there's a big difference as to how the DNA is delivered to the plant in order to initiate an infection. So it's a big difference between you touching some surface that's contaminated with a virus and having the virus stuffed into your mouth versus, AIDS doesn't really transmit by just drinking it, etc. I mean, you have to actually like do way more in order to get the virus into your body. And the same sort of also applies with the, yeah, the leaf curl virus is that it needs to get into the plant with the whitefly most of the time. Now we use agroinoculation, and that's where it's basically just use bacteria that normally inserts its own genes into the plant in order to have forced the plant to become, well, essentially a kind of plant cancer that just grows out and produces a compound that bacteria eats. So when you see little bulbs on trees occasionally, and quite often actually, that's caused by some of these types of bacteria that lives out in nature, forces the plant to grow these bulbs, and then lives there more or less and spreads out that way. Very common. And then you're just, of course, when you create your synthetic virus, you insert it into these bacteria. So the bacteria then inserts the virus into the plant. And then it becomes even more complicated because, like, depending on where you inoculate the plant and what life stage you inoculate the plant, that will infect the amount of plants that becomes infected. And one of my works I've been struggling a bit with here was optimizing that process to figure out, like, we use and different kind of Mycobacterium strain. We optimize as to using younger plants and then injecting it into the stem. That is for the case of the Agrobacterium and the virus method, way more likely to initiate the infection than other approaches. And this means when we have this set up, we can basically get to a point where we're looking for a very binary approach to infection. It's also better than the whiteflies because the whiteflies and well, you can't really see how much the whitefly actually manages to suck on the plant. So with this method, we actually know what's happening now. And that is also therefore superior to actually checking and screening for a particular mutation that would be resistance causing, because now you have 100%, okay, the virus is going in all the time with this approach when we optimized it. So we can say, okay, now it's going in 0% of the time. So we don't have to take a very large amount of plants in order to figure out, okay, definitely infected this time around. So you want to have like 100% what we call disease incidence when you expose the plant to the virus with the infection until we get plants that are actually having the virus develop. So that's basically the work with the agro-infection is optimizing that approach. I wrote a whole review on it because it sort of relates into a lot of also transformation of plants, of trying to get genes into plant. And they actually, the Gimli virus is a kind of a excellent study of this. They were also like some of the first things people did incidentally with wheat where they had a Gimli virus. They showed, oh, we can actually get any kind of DNA transfer happening to it because the Gimli virus starts up. But other viruses don't that start up that easily. There's definitely a mechanism, and it doesn't relate to how severe they are or how much they spread by the whitefly necessarily. Like some of those viruses can be very easily spread by the whitefly sort of feeds enough on the plant, then the white virus will sort of overcome the initial resistance and then they will stick into the plant. Some big one viruses actually just live in plants and just, yeah, propagate with the plants more or less indefinitely. But
Okay, I mean, very interesting. Yeah, and so you shared some of the preliminary results already now with us in this podcast, but is there something that we should be looking out for? Any publications you have coming up at NRI on your work in vertigation?
yeah, so of course we've submitted and got rejected on the first go because we just aimed a little high and they thought like, well, they've all, because people had already found out sort of way back that it was the trap Wen region that was doing it between beans and tobacco, and then like, well, then it's not novel that it's the part of region and all your discussion about it happening for tomatoes and cucumbers. Well, I disagree, and then like one thing a specific mutation out, and we did some more work trying that and writing in for a new journal, finishing the review, trying again here. Now we have and then meanwhile we've sort of been focusing up on seeing if we could also, because we have time, find the plant interactors to it. So because now we've figured out is this minimal region, and yeah, we get some results basically that it's doing multiple things. And it's basically kind of complicated to explain simply, but the essence of it is that we figure out that this sort of small part of the virus, I'd reckon it's sort of in a part of a virus. That's how to explain it. It's doing things that interacts with a lot of transcription factors in the plant. So it's probably what's doing it, figured out. And we basically found two different differential interactions between plant proteins for both the genes. Because the problem is the 60 base pair region I found and that's responsible sits in the part that codes for two genes. So the virus, because it's so small, basically uses overlapping reading frames to produce two proteins for it. And I found that it's doing the same thing in cucumber and tomato, right? But that little part is basically losing an interaction with a protein in both plants. But that, of course, is a bit more complicated now because now you have to consider, well, how does each plant respond? Particularly considering that we now know that we have an entire other component that's responsible for the spread of the virus being in cucumber, right? So that part is also necessary for it to be in cucumber. And now you have two interactions that's different, while at the same time, you know, there is an enormous amount of interactions in the DNAB component that's also necessary. Because the people already know what these are interacting with. The question is, are they differential between tomato and cucumber? And when you then now, because the difference part is, and the part of the conclusion is that this host jump is more complicated going from tomato to cucumber, basically, because it involves the DNAB. And then they're just losing the tomato infectivity by some point mutation interaction. Or they're interacting with something else because the plant will also have, if it interacts strongly with something in the plant, then that plant might have an easier time detecting the virus. And that, of course, goes into a very complicated discussion regarding plant immunity, which is kind of complicated because there's like many different genes. And then you go into the, well, we don't actually really know all the detailed conclusion because what happens is, yes, this pathway works like this, but then we also have the suppressors, the virus produces proteins to counteract this mechanism. And then sometimes it still works anyways, because then the plant have backup mechanisms that works anyways, and the virus have made backup mechanisms to counteract the plant doing it, and so these things have worked in lockstep, and we don't really know the bottom to that rabbit hole. So we just end up finding that, yeah, we find differential interactions between that is this little 60 base pair is responsible for, but it would be much more work to try and unravel how the whole thing would work together within the plants. It would also require a lot more work to try and recover additional unknown interactions. Because so far my work has only focused on all the known interactions. The people already studied these proteins quite extensively. They already found out what they're interacting with in the plant, they think. Then the question comes, well, because different methods of finding interactors might fail in some regards and not get all the correct interactions. So then the question comes, well, if I start redoing it, I need new methods to try and look for new unknown interactions. And that becomes, well, we're trying to do something unknown for something unknown. And that's a big ask, particularly considering that we initially were just aiming at figuring out what the virus was doing. And we did that. And we just had spare time and we figure it's doing a lot of stuff. It's doing multiple things at the same time.
Okay, I mean, very interesting, and still, as you sketch it out, quite some research left to be done in that area. Finally, I mean, projects like Vertigation, collaborative projects are obviously important in fighting these emerging viruses like tomato leaf curl New Delhi virus. How has the NRI benefited from being involved in a project like Vertigation?
No, well, so it sits well within the NRI remit of studying diseases, understanding their integrated pest management, which I'm basically not so much part of, but also because that's sort of more in terms of like how you put plants out, and how you have resistant varieties, limiting it and keeping them away from the insects, etc., and the insects away from the plant. So that's like one thing, and that's sort of, but it fits. Of course, the main benefit is, of course, that we're starving off unemployment for another 2 years, and we kind of solve out the main question of like figuring out the host jump of these viruses, which of course how is this useful to know the host jump mechanisms? Well, kind of in a way that we now sort of can realize that the viruses we have that infect cucumber are most likely going to be able to quite easily adapt going over to tomatoes, or just regaining whatever made them unable to infect tomatoes. Whereas some of the viruses that are staying within a group of viruses that infect tomatoes are very unlikely to grapple and become significant in cucumbers. And that in reality, you have a subgroup of viruses that are bipartite, because you can look in the phylogeny between them, and that these viruses are likely going to be infecting different sorts of cucurbits, cucumber melon, etc., with different efficiency. And that, from there on, it's just something sometimes, because here's the other problem, it's like different variants of plants, cultivars, and variants of viruses might be changing the difference between being able to infect and not infect them. And like we got one of the new viruses that's sort of a bit off. It also allows us to do some little extra research here where we got like figured out, okay, in reality, what's probably making the cucurbit virus that's right now becoming problem out in India. So, and because we see the tomato leaf curly jumped over to Europe, it's probably gonna come over as well. And it's sort of also, that's also part of this here, is understanding like how the viruses move around, seeing them emerge. And that's sort of that the virus becomes more severe because of its better ability to probably synergize with other tomato leaf curl viruses. That is, the viruses have a very complicated means. And that's actually some of my bonus work I've also been looking into, because I figured this out sort of random, is the mechanisms of the virus being able to synergize or not synergize with each other. Because sometimes you can have a virus infect a plant, and then you can't have two viruses of the same sort in the plant. Often you'd find that some viruses make complexes of viruses of different sorts, going even to RNA and DNA viruses, both being in the same plant, making it more severe. But also, some groups of viruses can infect the plant together, and some cannot. So two of the viruses we have cannot infect the plant together. One virus that is severe will just kick the other virus out. If it's already infected with a weak virus and the strong virus comes in, it removes the weak virus. Whereas one of the other strong viruses, if it comes in and the weak virus is there, well, it will make slightly more of the weak virus as well. So now the weak virus can move on, even if that's present, non-present. So those kind of dynamics are also taking place. So that's sort of some bonus we figured out, because I just decided to randomly put two viruses into the same plant and start seeing what happens. And then that turns out to be a big, like, wow, that's going to be really interesting. On top of it, because we basically had now figured every single host jump part there was to it.
That was the second edition of the VIRTIGATION Plant Health Stories podcast. Make sure to subscribe on our Spotify Plant Health Stories channel in order to stay tuned for upcoming editions. The VIRTIGATION project is a Horizon 2020 project funded under the grant agreement number 101000570 by the European Commission.